Hello. Uh, in this video, we're going to see how cells actually make ATP from photosynthesis. I've already alluded to it. You know, they move the electrons and along some electron transport chain in the thylakoids, in the thylakoid membrane, and that's how they get ATP. So let's look at all these topics. All right. So let's start with photosystem 2, assuming that you know, you are dealing with a green plant that has photosystem 2, splits water, generates oxygen. We don't see this in this reaction. The electrons will find their way onto Q. Okay, but we put a P in front of the Q, so the ubiquinone, because it's the plant version, but it's very similar. So Q again, so PQ, let's call it, is the membrane-bound factor that moves around from, and it's exclusively in the inner portion of the membrane. Now, when Q receives these electrons and gets reduced, it picks up 2 protons from the stroma. Okay, remember the stroma is the outside. So then, okay, just like we had seen in mitochondria, the point there is an electron transport complex here that will take take these electrons from Q. So Q will be reoxidized again and puts them onto the soluble version of cytochrome C that we had seen, and now that can then take it on to the next one, right? So in the process, again, the 2 protons will find their way into the lumen. So we had done the Q cycle, you know, when we drew it on the board at the time with a mitochondria. It's exactly the same thing here, except we're dealing with the plant versions. So this is a more beautiful schematic, right? We have the reaction center, you know, we have the antenna complex, the photon hits there, and then moves on, as we had seen with the antenna, it hits the reaction center. That's photosystem 2. The electrons will find their way onto Q, and from Q, you know, we have the cytochrome complex, and they keep going. So you have now protons that are getting into the lumen, the thylakoid lumen, and all you have to do is really put an ATP synthase. Okay, and now the protons want to go out. That's the proton motive force that you have built. You have charged your batteries in the thylakoid, in each of the little thylakoids in the chloroplast, and as the protons go out, you make ATP. Okay, so it's exactly the same principle. And in fact, I had shown you this slide from ATP synthase that Andre Jangendorf had shown that all you need is a pH gradient, and you can make ATP as long as you have thylakoids. And the experiment is super simple. You have the thylakoid here, and shown here with very few protons inside. Okay, so the pH is so it's a closed vesicle, if you wish. But of course, it's not pure at that point. When he did the experiment has been done with pure membranes with just ATP synthase on them. But at the time that Jangendorf did it, it was just membranes purified from a chloroplast. And so the question is, how do you put protons in, right? Well, you just put it in a pH 4, so low pH. You wait some time, and eventually, several hours, sometime, and eventually protons will leak in, okay? And then, so you will have plenty of protons in, and then all you have to do is just put them back out on a low proton solution, and the protons want to go out. If that's how they make ATP, if it's the proton translocation that actually makes ATP, well, then you should get ATP, because these membranes also have the ATP synthase on them. He didn't do anything with ATP synthase, so it left it there. And sure enough, he saw ATP production, and all he had to do was just change the pH. It was a beautiful experiment, and then it was done in an even better way to have a pure membrane, just stick ATP synthase on it, so you don't have to worry about any potential other impurities that may have been on the thylakoid. And that's a proof that all you need to make ATP is not some, you know, series of oxidative substrate level phosphorylations that people were looking for. Right? That's all you need is a charged battery, and that's it. Beautiful. Now, what about the electrons, right? So fine, we get ATP, but what about the electrons here? What happens to them? So when you have this photosystem 2 and photosystem 1, the electrons will actually end up on photosystem 1. So you start from photosystem 2, you make the oxygen, the electrons move on, you make some ATP, and the electrons actually will move now to photosystem 1. And 4 more photons, 4 more flashes will hit, okay, and they will be raised up. And then you're going to have another electron transport chain, and then finally you're going to find them onto ferredoxin. Ferredoxin is a cool molecule because you can actually reduce an ADP and into an ADPase, right? Oh, and if you do it this way, you have captured essentially 38% of the energy in ATP production and ADPase synthesis. So what's the outcome of photosystem 2, photosystem 1? The non-cyclic photophosphorylation, as it is called. You'll see why it's called non-cyclic. It's also called oxygenic because you make oxygen. But the real, oxygen is a byproduct here for the plant's perspective, okay? What the plant cares about from this process is to make reducing power and energy currency. And it got them both. It got ADP and an ADPase. That's the view of this electron transport of photosystem 1, where you have the extra electron transport onto ferredoxin. So really what we have here is photosystem 1 being a powerful reductant. It transfers electrons to ferredoxin. It's not easy, okay, to do that, but so you need all the energy from light to do that. And once you have, so that's where this is who pays eventually to reduce ferredoxin. It's the sun, the light. And ferredoxin can now actually reduce an ADP. And why can it do it, thermodynamically speaking? Because it has an E value for the half cell reduction that's even more negative than the minus 320. So to actually reduce an ADP to an ADPase or an AD to an ADH, you need to have something that's even lower, okay, than minus 320 millivolts. And in fact, the half cell for ferredoxin is lower. So it's about 100 millivolts. So it's a plus. So it's thermodynamically favorable. It's about 100 millivolts. So it happens. But again, who pays for it? That's the photo energy right there. That's one option that ferredoxin has, however. It can transfer them back to Q. And why would you do that, you may ask? Okay, so, well, ferredoxin can pass them on to Q, so the electrons get passed on to Q, so it reduces Q. Okay, ferredoxin can reduce Q. And then the electrons pass on through the cytochrome complex. You have the proton pumping, and now you can actually make ATP if all you engage is photosystem 1. So here is a case of photosynthesis where you don't make oxygen, okay, because you do not engage photosystem 2. You just play around with photosystem 1, and sometimes you pass on them, and all the time you pass them on to ferredoxin. Then ferredoxin can either pass them on to an ADP to make an ADPase, reducing power, or pass them back to Q, and then you get ATP out of it. So that works too, okay? So you got, from a plant's perspective, remember, the plant doesn't care about oxygen. It's a byproduct that we care about, but all the plant cares about is reducing power and energy currency. And just with photosystem 1, you can get both outcomes achieved, all right? It is, that's the view of simplified view, the same thing. If you just have photosystem 1, and we know that there are photosynthetic organisms that all they have is photosystem 1. How do they survive? How do they do it? They do it just like this, and it's all cool. And that's called cyclic photophosphorylation because, of course, you go around and around. There is no oxygen release, as we have said, and it's less efficient. 12% of the energy is captured in ATP in this case. Now, and of course, green plants can do both, or they can just do photosystem 1. They have all these options, but the way the photosystems are drawn like this, you have 1, and then you have 2, and so on and so forth. They never work in real life. The reason is, if they are too close to each other, okay, you can short circuit the whole system. So how do plants avoid that? Well, you put them far away from each other. Photosystem 1 is in the unstacked lamella, okay, somewhere there, for example. And photosystem 2 is in the stacks that we saw earlier. Well, but you might say, how do they communicate then, all these electrons? Well, because you only rely on these little guys, plastoquinone Q, so Q and cytochrome C, they can move around one in the lipid bilayer, and the other one in the aqueous phase touching the membrane. So that's how it can happen without short circuiting. So the whole going from photosystem to photosystem 1. Finally, we're going to close with photosystems, with actually my favorite, because it's really the simplest, most beautiful thing, and illustrates the whole point. You don't need any complex stuff. So here the reactive compound is retinal, right? And of course, we're talking about bacterial rhodopsin. It's the simplest light-driven proton pump. So that's retinal at the heart of rhodopsin. And it's a very simple compound. It looks like this. It's all in the all trans configuration. All the double bonds will be all trans, but you shine some light on it, and then you flip one, and it flips to the cis configuration. Now, if you have this compound now into a protein, moving essentially an arm, from the cis to trans, you will expect that you will cause some conformational change. If you can couple this conformational change with a translocation of protons from one side of a membrane to the other, then what you have? You can charge a battery, right? And in fact, bacterial rhodopsin is very simple. It's just a bunch of alpha helices, and you have the retinal in the interior. So as that flips by light from the cis to the trans configuration, you can see how it can push around some of these helices, and then you have proton translocation. And so if you put bacterial rhodopsin on the membrane in a bacterium, this is Halobacterium salinarum, you put it in the periplasmic membrane right there. So the protons will pump out into the periplasm, and then of course they want to come in. So, and as they come in, all you have to do is just stick an ATP synthase, right? And then you got your ATP in the cytoplasm. It illustrates the whole point. And again, the glory of the chemiosmotic hypothesis by Peter Mitchell, because all these phenomena, they're really the same thing. You can have simple things like rhodopsin or the complex electron transport chains, but it's really the same fundamental principle. It's one of the best examples of unifying biology, if you wish. All right, so, and this is an artificial one that doesn't exist in nature. Many photosynthetic organisms exist, but most are not, like yeast, my favorite one. Can you make it? Well, if everything I've been telling you is true, all you need is to have something that, is photosensitive, raise the energy of some electrons, and then use that to pass them on to, let's say, NADP and make NADPH. And a few years ago, again, it was published this incredible paper in a prominent journal in Science, where people took yeast, and all they did is just stuck some iridium. It's almost like a rare earth thing, and it's used in chips, electronic chips, and you put it on the membrane, or on the surface. You just coat Saccharomyces cerevisiae yeast cells with that stuff, and then you shine light on it. Oh, and by the way, at the same time, just to demonstrate the proof of principle here, you have eliminated the ability of yeast to normally make NADPH, which we will see how it makes it, through the pentose phosphate pathway. So it can make NADPH, unless it uses photosynthesis to reduce NADP to NADPH. And in this case, how will it do photosynthesis? Again, it's with the artificial chip-like indium, not iridium, indium phosphide. And then miraculously, these people found that actually yeast could use these high energy electrons to reduce NADP plus to NADPH. And as a readout, they had this reaction that totally depends on NADPH. And we're going to see this reaction when we discuss synthesis of aromatic amino acids to make this compound. And this is all, and that's actually a very valuable compound, and it's all free in a sense. Yeast can make it if you shine light, this particular yeast. And so it does have actually, a biotechnological significance, because you can make a valuable compound for free, right? So this is an artificial photosynthetic system here. And with this, we close the photo part of photosynthesis. We got our chemical energy in the form of ATP and NADPH. And now in the next videos, we're going to see how we put them to use to synthesize the goodies, glucose. And we'll stop at this point.